Hi everyone. In this video I am going to explain about stem anatomy under this transfer section of monocotyledonous stem. Internal structure of monocot stem. The internal structure of a typical monocot stem shows following features. Under this, the very first one is epidermis. Epidermis is the outermost layer of monocot stem. It is uniseriate or single layered. Multicellular hair, which are called trichomes, are absent. Stomata also very less in number or may be absent. Outside of epidermis, a layer is present which is made up of cutin is called cuticle. Epidermis plays a significant role in protection of stem. Remember, in monocot stem, epidermis is as usual single layer, outermost layer. But trichomes are absent, stomata is less in number or absent, cuticle is present. The main role or the main function of the monocotyledonous stem epidermis is protection. Second one, hypodermis. In monocot stem, hypodermis is 2 to 3 layered and just present below to the epidermis. The hypodermis of monocot stem is completely composed with sclerenchyma cells, and the main function of this hypodermis is providing mechanical support to the stem. Third one, ground tissue. The entire mass of parenchyma cells next to hypodermis and extending to the center is called ground tissue. There is no differentiation of ground tissue in monocot stem, but peripheral side cells are smaller ones and central side cells are bigger in size. Here you have to remember hypodermis of monocot stem is completely composed of sclerenchyma, and function of this hypodermis is giving mechanical support to the stem. Coming to ground tissue, it is completely composed of parenchyma cells. These are present up to the center. And one more thing, the parenchyma cells of ground tissue which are situated peripheral sides are smaller ones, which are situated central side, those are bigger in size. Fourth one, vascular bundles. Many vascular bundles are present in monocot stem. Each vascular bundle surrounded by a sclerenchymaceous bundle sheath. All these vascular bundles are arranged in scattered manner in ground tissue. That's why this type of vascular bundles are still called atactostele. In monocot stem, vascular bundles are generally oval in shape. Each vascular bundle is conjoined, which means xylem and phloem both are arranged in same radius. Collateral, xylem and phloem are in 1:1 ratio, and closed, which means there is no differentiation, there is no cambium between xylem and phloem. Closed vascular bundles does not show secondary growth. Here we have to remember peripheral vascular bundles are generally smaller than centrally located vascular bundles, which means peripheral vascular bundles are small in size, central vascular bundles are bigger in size. Remember, vascular bundles in monocotyledonous are large in number. These vascular bundles are surrounded by sclerenchymus bundle sheath. All these vascular bundles are arranged in scattered manner in ground tissue, which is called atactostele. The peripheral side vascular bundles are small in size, and centrally located vascular bundles are big in size. All the vascular bundles are oval in shape. Each vascular bundle is conjoined, collateral and closed type of vascular bundles. Let's see the diagrammatic representation of vascular bundle, which is in round or oval shape. So these vascular bundle covered by a sclerenchymus protecting sheath, which is called bundle sheath. Within that, they contains conjoined, collateral, closed type of vascular bundles. Here xylem and phloem both are arranged in, are present in same radius. but there is no cambium between xylem and phloem. That's why these are called closed type of vascular bundles, which does not shows secondary growth. Under vascular bundles, we have to know xylem and phloem. Xylem. In xylem, number of vessels are very less. In metaxylem, there are two large xylem vessels are present, while in protoxylem, there are only one or two but small xylem vessels are present. Xylem vessels are arranged in V or Y shape in vascular bundles. Just beneath protoxylem vessels, there are a water cavity which is cyzo-lysogenous in origin, but major part of water cavity is lysogenous in origin. This cavity is formed by disintegration of the element present below the protoxylem and neighboring parenchyma cells. Exception: there is no water cavity and bundle sheath in the plant called asparagus. The next one is phloem. Phloem consists of sieve tube elements and companion cells, but phloem parenchyma is absent. The next part is stele. Atactostele is found in monocotyledonous plant. Atactostele in the sense the scattered arrangement of vascular bundles in ground tissue of monocot stem is called atactostele. Atactostele is the most developed stele in plants. Let's see the diagrammatic representation of transverse section of monocot stem. See, in transverse section diagram, the outermost layer is epidermis. On the epidermis, cuticle is present. Below to the epidermis, there are 2 to 3 layers of sclerenchyma cells which are called hypodermis, which gives mechanical support to the stem. Below to that, parenchyma cells are there which are called ground tissue. In the ground tissue, several vascular bundles are arranged in scattered manner, which is called atactostele. So vascular bundles are conjoined, collateral, closed type. See, the next diagram is outline or ground plan of transverse section of monocot stem. For your better understanding, let's check these diagrams carefully. Here, the very important thing is the differences between monocot stem and dicot stem. Coming to epidermis, the epidermis of monocot stem is generally comparatively smaller cells, but coming to dicot stem, epidermal cells are large cells. In monocot stem, hair or trichomes are generally absent, but in case of dicot stem, multicellular hair or trichomes are present. In monocot stem, hypodermis is sclerenchymus, but in case of dicot stem, hypodermis is collenchymus. In monocot stem, cortex is absent, but they have hypodermis. But in case of dicot stem, cortex is made up of many layered parenchyma cells, and cortex differentiated into hypodermis, general cortex, and endodermis. But in monocot stem, endodermis is absent, but in dicot stem, endodermis is present. It is called starch sheath. Pericycle is absent in monocot stem, but pericycle is like semilunar patches in dicot stem. Medullary rays are absent in monocot stem, but medullary rays are present between vascular bundles in dicot stem. Pith is absent in monocot stem, but pith is present in dicot stem. Coming to vascular bundles are scattered in monocot stem, but vascular bundles arranged in ring-like structure in dicot stem. Vascular bundles are conjoined, collateral, and closed type in monocot stem, but in case of dicot stem, vascular bundles are conjoined, collateral, and open. There are differences in the size of vascular bundles in the center and at periphery in monocot stem, but almost all vascular bundles are same size in dicot stem. Vascular bundles are oval in shape in monocot stem, but in case of dicot stem, vascular bundles are wedge shaped. In monocot stem, bundle sheath is covered by sclerenchymus bundle sheath, but in case of dicot stem, bundle sheath is absent. Phloem parenchyma is absent in monocot stem, but phloem parenchyma is present in dicot stem. Up to here, the stem anatomy, both transverse section of dicot stem and transverse section of monocot stem were completed. In next video, I will explain you about leaf anatomy. Thank you.